So, to first illustrate, the importance between the microbiome and the immune system, we really have to understand the relationship that we have with organisms at a high level, right? So a human is actually a holobiont. A holobiont is a superorganism. So a human is basically a collection of bionts, of synbionts, if you will, meaning numerous organisms working together in concert to promote the health and the wellness of the whole. And so we are a superorganism. We're similar to a walking, talking rainforest. So we're made up of thousands of different species that all work together in this container that we call the human body. And when you look at the human system from that perspective, it starts to become clear, or at least evident to some degree, that microbes and our own cells need one another in order to function properly. Keep in mind that our immune system is there to protect us primarily from microbes, all the while being completely covered and surrounded by microbes, right? So it's a difficult task for the immune system, and we'll peel back how the immune system actually does that with the help of the microbiome. This is something you all know very well, the basic kinetics of the immune response. Of course, we've got the early stage response, which happens in hours when something enters in. That's the innate immune response. And then you've got the late innate response, which is typically facilitated by dendritic cells and macrophages. And then we move on to our antigen presentation of the adaptive immune response, which of course involves B cells and T cells. And hopefully at the end of that, you end up with immunity, true immunity, a long-term protection. And of course, the time of response goes from hours to days in that manner. The thing that's really important to note, and this will become an important theme in this whole lecture about the role of the microbiome, is that inflammation is a key part of the innate immune response, right? We need inflammation for the innate immune system to function properly. We require that inflammation, and you'll find out exactly why, but you already know some of the areas of inflammation that are required. I mean, of course, the part of the inflammatory response is kicking up the production of fever and heat shock proteins and quarantining infections into certain areas and so on. But there's other really important aspects of inflammation that is controlled by the microbiome itself. Now, as we start progressing away from the innate immune response towards the adaptive, we actually need a very important anti-inflammatory step. And if we don't get that anti-inflammatory step and the innate immune system is allowed to run for longer than it should, then we start getting diminishing return from the immune response. We start getting to a place where the immune system itself is harming the host more than the actual pathogen, right? And we know that even in COVID, as you look at things like the cytokine storm and the people that go into respiratory distress and so on, these people are more often being harmed by the immune response more so than the actual virus itself. And of course, if you look at both the early innate and then the late innate response, all of these processes will require the microbiome in virtually every step of the way. And dendritic cells and macrophages are a critical part of this whole escorting of the function of the immune system from that early illness, that early innate response, to the late innate, all the way down to the adaptive. And so the early innate response moves down to the late innate response. Of course, there's inflammation in this process. Then you've got the early adaptive response that kicks off as you start antigen presenting and as you start proliferating, B cells and T cells in the lymph nodes. That also requires an anti-inflammatory step to slow down the damage that's occurring from the innate immune response. And then hopefully after that, you end up with more long-term adaptive immunity. Now, every single step in this process requires something from the microbiome. And that's the really important thing to note, is that we've got a specific immune kinetic response, right? And if anything within this stepwise manner goes wrong, the immune system fails to respond as it's supposed to. And so it's really interesting to note that this really important system that's in charge of protecting us as the host requires the microbes within our system to help facilitate the transformation from one step to the other. Right. So we'll talk about what some of those steps are, how specifically does the microbiome do that, but just keep that in mind as an overall theme. And then even when you look at the anatomy of the immune system, you've got the secondary lymphoid organs and tissues, things like the Waldeier's rings, of course, the lymph nodes, the lamina propria, the Peyer's patches, mesenteric lymph nodes, and all that. And then, of course, you've got the primary lymphoid organs, the thymus and the bone marrow, which is really good at producing immune cells, right? T cells and so on, all the white blood cells, they're all produced in these primary lymphoid organs, but they're produced in a naive state. Those immune cells that are being produced by our primary lymphoid organs do not know how to function as immune cells just yet. They have to go to the secondary lymphoid tissues and organs to get tutored, if you will, and they have to get matured and proliferate in those secondary tissues. And all of those secondary tissues are connected to the microbiome, and the microbiome has influence over all of them. And we'll touch on some of that as well. So the microbiome plays this important role in tutoring and helping proliferate the naive immune cells that are being made in the thymus and the bone marrow. Now, most of the immune action in the body occurs in the mucosa. As you guys know, the mucosa covers the vast majority of the inside of our body, right? And the largest surface area of the mucosa is in the digestive tract itself. So the mucosa in the digestive tract has about 400 square meters in surface area. Compare that to the skin, which is about 2 square meters, and 400 square meters. I can't remember if in the Philippines you use the metric system or the weird system that we use here in the States. But if you use a metric system, then you'll understand what 400 square meters is. If you use I think it's the empirical system, it's about 4,000 square feet, which is a massive apartment by any standards. All of that mucosal surface area is folded up in the body and covers virtually every entrance in the body. It's the largest portion of the immune system. Most of it is, of course, found in the gut, in the digestive tract, and it lines every entry area into the body, whether it's through the respiratory tract, the digestive tract, even the urogenital tract, even going through the skin, you're going to hit a mucosal layer at some point. And so it becomes the largest site of immune sampling in the body, because almost everything that enters the body enters through the mucosa. And in the mucosa, the decisions are made as to how to respond to this thing that's entering the system, right? Whether it's a virus, a bacteria, a fungi, a toxin, a food, whatever it is, the decision to respond or not to respond or how to respond is made in the mucosa. And because that decision is made in the mucosa, and the largest control mechanism and sampling tissue of the mucosa is in the digestive tract, the gut kind of becomes the central command center for all of the mucosal tissue in the body, right? So if the gut is dysfunctional, and if the gut mucosa is not functioning properly in terms of sampling, building oral tolerance, identifying viruses and pathogenic organisms in a proper way, then the rest of the mucosal tissue will get the wrong message and will get the wrong feedback and start to act in an inflammatory manner as well. That's how a dysfunctional gut can affect acne, for example. And we've just completed our second study in acne. That's how a dysfunctional gut can enhance chronic sinusitis. That's how a dysfunctional gut can drive chronic bacterial vaginosis, because a dysfunctional gut will affect the rest of the mucosal tissue. And then, of course, the mucosal system is covered with microbes. That's where most of the microbes in the body live. They live in the top layer of the mucosal tissue. Of course, the GI tract has the largest amount of microbes living in that top mucin 1 layer, the closest to the lumen. And so the immunobiology of the mucosa becomes extremely important, but trying to understand why for that second claim I made about the microbiome being the eyes and the ears of the immune system, in order to try to understand that, we have to understand how this mucosa is actually structured and looks, right? So if this is a sample of the mucosa, the microbes in there would look very similar to this. They're sitting shoulder to shoulder, cell membrane to cell membrane, cell wall to cell wall. the mucosal system is covered with over 40 trillion microbes, right? So it's 40 trillion microbial cells in that 400 square meters of surface area. So they're all sitting there. It's an extremely crowded place. Now, compared to that 40 trillion microbes that exist, there are only about 200 million immune cells whose job it is to survey this entire surface area, right? 200 million immune cells to survey an area that is 400 square meters that is covered with over 40 trillion microbial cells. That is a 200,000 to 1 ratio of immune cells to microbial cells, right? So that's a mind-boggling task for the immune system, because each of those immune cells is responsible for trying to figure out which bacteria, which virus, which fungi is friend, which is foe, right? And it's a 200,000 to 1. That means for every 200,000 microbes, for every one immune cell, they have to monitor a surface area that's large enough to contain over 200,000 microbes. And so that becomes an arduous task for the immune system. And the big question would be, how does the immune system actually do that? And then how does the immune system recognize friend from foe when viruses, bacteria, and all that can look quite similar, right? We can identify the differences by doing genetic analysis, but we know that the immune system is not running genetic analysis. So how can they do that? And for that reason, if you look at a system like this, right, imagine that this was a festival and there were 200,000 attendees at the festival, and it was a closed festival, closed off with gates, and you were the only security guard in this festival whose job it was to protect all of these individuals from any egregious players. And then you know that among the 200,000, there's only one individual that actually means harm to the people there. So the 199,999 people are perfectly fine. There's one individual that means harm. And let's say that individual moves around and is spread around. There is no possible way for a single security guard to monitor those 200,000 people to find the one individual that potentially is causing a problem. The only way that it's possible is if the other 199,999 people were also working with the security guard and were radioed in to him or her. And if they saw anything unusual, they would call you and tell you exactly where they saw it, right? So that is a neighborhood watch type of mentality. That's exactly what's happening in the mucosa. The vast majority of commensal organisms in the mucosa are keeping an eye out for changes to that ecosystem by a new egregious player, right? A new infectious virus or infectious bacteria or infectious fungus. As those enter into the picture, the first cells to pick up the change are actually the commensal microbes in that region. And they are acting as the neighborhood watch or the eyes and ears for the immune system in order to respond to the presence of that new pathogen. Now, this happens through a number of crosstalk mechanisms. I promise I'm not going to go through all of these at all. I'll just point out a couple simple ones like the production of short-chain fatty acids, which are postbiotics. These short-chain fatty acids like propionate, butyrate, acetate will bind things like G protein couple receptors on the intestinal epithelial cells or on the intestinal, the enteroendocrine cells that are within the epithelium. That kind of binding can actually create signals to turn on genes to improve intestinal adaptation and improve barrier function. It can enhance goblet cells production of mucus. It can also activate dendritic cells or macrophages or prevent inflammatory responses among dendritic cells and macrophages. It can help differentiate T cells from naive T cells to T regulatory cells to improve oral tolerance. There's lots of different ways in which the microbes in the mucosa influence the immune response through crosstalk. And it's that crosstalk that is critical for the microbes to be able to alert the immune system to the presence of something new, right? So let me give you some actual examples of how the commensal microbiota plays a role in fighting viral infections. And these are common viral infections. So for example, during a norovirus infection, the lactobacilli, the commensal lactobacilli in the GI tract and other commensal bacteria will trigger the release of interferon beta and interferon gamma when they detect the presence of the norovirus, right? So as the norovirus is coming in and infecting the human host cells, the local lactobacilli will pick up that change in that environment, and they will trigger the release of interferon beta and interferon gamma. And as you guys know, interferons can be really potent anti-replicative compounds against viral infections. And this also then alerts the innate immune system to the presence of the virus, and so that the innate immune system can be recruited to the site of infection. Something like vitamin A, for example, a micronutrient, will provide the substrate for the commensal bacteria to make these interferons. And that's how some of these micronutrients become important as well. And then take a rotavirus infection. Bacterial flagellin from commensal bacteria that have flagellin will activate the expression of certain pattern recognition receptors, and this will trigger the expression of toll-like receptor 5, which then stimulates the release of interleukin 22 and interleukin 18. So while the rotavirus infection is progressing and ongoing, the bacterial flagellin from the commensal bacteria is trying to stimulate the release of interleukin 22 and 18. And when you look at what those two things do, it becomes extremely clear why they're so important during a rotavirus infection. For example, interleukin 22 helps repair the damaged epithelium, because as the virus is infecting epithelial cells, those cells get damaged, die off, and leave big gaps in there. So we need to repair that damage that occurs so you don't have big spacey gaps in the intestinal epithelium. And then the interleukin 18 also induces apoptosis in infected epithelial cells, so they stop becoming viral factories, right? So the bacterial flagellin is not only stopping the viral factory, but it's also stimulating the repair of the damaged epithelium because of that cell that's now dead that was infected. Bifidobacterium breve, and especially in combination with the galacto-oligosaccharide and fructo-oligosaccharide, have been shown to prevent rotavirus infections, and in fact doing so by increasing interferon gamma, IL-4, TNF-alpha, and toll-like receptor 2. All of this increases mucosal immune defenses. Commensal bacteria also produce short-chain fatty acids, like I mentioned, which is required to increase and maintain mucus production, because butyrate, for example, is the main fuel for goblet cells to make the mucin layer. And this creates that strong barrier against pathogens, because it's harder for the pathogen to get down and infect the intestinal epithelium or any epithelial cells or endothelial cells if they have to make their way through a mucosal lining. In addition, these commensals also increase the synthesis of antiviral compounds, things like reactive oxygen species and defensins. And defensins are extremely powerful antiviral compounds and can be antibacterial to some degree as well. And these all will prevent local viral replication. During influenza infection, commensal bacteria can trigger the release of inflammasomes, and inflammasomes are the major pathology by which we fight off influenza infection. And this potent defense system against influenza will induce dendritic cells to migrate to the local tissue, local lymph nodes, to stimulate influenza-specific T cells to respond then into the gut, and shuttling the response to the more potent and less damaging early adaptive response. So we're trying to get the adapted T cells or the B cells to activate, and the inflammasomes do that in a significant way, and those are being activated by commensal bacteria. Other examples are gut microbes. They regulate the respiratory mucosal immune response in the influenza infection, and they do that through stimulating IgA secretion, Th1 activation, cytotoxic T cell priming. We also know that the influenza virus, when it's present in the lungs, the gut commensal bacteria tend to increase the presence of innate immune cells in the lungs by causing the release of cytokines like IL-33, IL-1 alpha, IL-1 beta, and so on. This release of cytokines causes more natural killer cells and dendritic cells and macrophages to end up in the lungs. But keep in mind, what we're talking about is the gut microbes, right? The gut microbes, the gut commensal bacteria can increase the presence of immune cells in the lungs, which means that there's some communication between the lung microbes, who are the first ones to detect the presence of the virus. Then the lung microbes are communicating with the gut microbes to indicate that there's an infection going on in the lungs. And then the gut microbes recruit the immune cells from that local region to go towards the lungs where they can fight off the infection, right? This gut-lung axis is a really complicated scenario. And in fact, we don't really understand most of how this communication happens between the microbes in the lungs and the microbes in the gut, and then how the microbes in the gut send the immune cells to the lungs. How does it direct it to the lungs? That's still not very well known, but we know it happens because you can study it happening, but it's hard to understand the communication tool. So there are things happening between the microbes and the immune system that we don't even understand just yet, but we know it happens. And when the virus levels are low, obviously, if you have influenza, you want nice, robust immune response. Inflammation is going to be a part of that response. But when the virus levels are low, gut commensals do the opposite by stimulating the release of anti-inflammatory cytokines like interleukin-10, so that the lungs maintain low levels of inflammation when it's being exposed to all of these irritants that are out there in the air, right? Because if the lungs responded with a robust innate immune response to everything that enters into it, people would have severe asthma all the time, right? And that's how asthma occurs, right? You've got this hyperactive airway, and so irritants, things that come in, whether it's allergens out there or toxins or it's exercise induced, all those things drive an inflammatory response. So to maintain healthy lungs and maintain tolerance for the things that your immune system in the lungs shouldn't be going after, the commensals increase IL-10 production when there's no active infection. So your lungs have good tolerance for everything around you. That's why we have an epidemic of asthma in the US alone. We have over 10 million kids with asthma, and asthma, the prevalence rate of asthma in the incidence rate of asthma, both going up quite steadily. And it's got to be because of these dysfunctional gut microbiomes. The balancing act is a great example of the gut-lung axis, like I mentioned. Another example is Staph aureus on airway surface. Will recruit monocytes that mature to macrophages, of course, and they do this through the activation of toll-like receptor 2 during the lung infection, and this leads to a reducing in the damage of lung tissue in the acute infection. So Staph aureus in the lung plays an important role in that response and minimizing the damage to the lungs. Respiratory commensal bacteria like corneum bacteria can modulate toll-like receptor 3 antiviral responses to the RSV virus by enhancing the production of TNF-alpha, interleukin-6, interferon gamma, all of these through increasing T-cell proliferation. And we know that butyrate, for example, from commensal bacteria, lower inflammatory damage post-early innate by activating the G protein coupled receptors on cell surfaces. I showed you that as an example in the schematic, where the key anti-inflammatory step during the immune response that allows the immune system to move into the adaptive phase of the response, in part is coming from the butyrate being produced by commensal bacteria. And if you don't have adequate butyrate levels, it becomes really hard to tone down that massive inflammatory response. We also know, for example, Lactobacillus crispacus, when it's dominant in the vaginal canal of women, this was a study done on South African women, it can actually inhibit or decrease HIV infections, even to women who have been exposed to HIV unprotected. Right. So that's how powerful that microbe is as a commensal organism. It can protect the host from this type of viral infection if it's there in the right proportion. Now, we know also that not only are the microbes doing these kind of mechanics to help detect the presence of infectious viruses or bacteria or any other organism, they're also providing the threshold signal for the immune system to respond at all. Right. So that was a really interesting finding between these groups of studies that happened over a course of almost 10 years. This first study here showing how commensal bacteria will calibrate the activation threshold of the innate antiviral immunity. Right. So what we don't want, is our immune system overreacting to everything, right? That's as bad as our immune system not reacting to things, right? So overreacting is equally bad as not reacting. And it becomes the job of the microbiome, as it turns out, to set this activation threshold, meaning at what stimulus level does the immune system actually start acting? And at what stimulus level and below does the immune system ignore it? Because that is part of the tolerance. If our immune system reacted to everything we came in contact with, we would constantly be sick. So what they've shown is that the data that indicated that commensal derived signals provide a tonic immune stimulation that establishes the activation threshold of the innate immune system required for optimal antiviral immunity. So when you've got a viral infectious agent entering the body, it becomes the job of the commensal bacteria to provide the activation signal to the immune system to react to that virus, right? That seems outlandish. It seems crazy that our protection, our system that's designed to protect us from infectious agents that could kill us, we've outsourced the activation and in large part the functionality of that system to microbes in our system. And so it's really bizarre. And that's why I mentioned earlier in the beginning that it's a beautiful, elegant example of symbiogenesis, right? We've identified that we need the microbes to act as our guards, if you will, our eyes and ears in this heavily microbe-based world. And because we've created that symbiotic relationship with them, they are now the ones that dictate how our immune system functions. So here's a follow-up study along the same lines, and there are some, I think, shared authors among this, but this is a study published in 2020 where they found that type 1 interferon, which is signaling from the microbiome, is the key signal that was shown to be required to tone and poise dendritic cells to respond to pathogen entry. Right. So in no-biotic mice that were grown without a microbiome, and there's no microbiome there to provide that signal, and even though the mouse has all of the immune components, all of the T cells and B cells and dendritic cells, macrophages and all that, if those if those animals were infected with the virus, obviously deliberately, their immune system would fail to respond. The dendritic cells would stand by and just watch the host cell get infected. Right, which is mind-boggling when you think about it. But then when you engraft a microbiome into those same animals, all of a sudden the immune system starts to respond to the presence of a virus, right? So the immune system would not even respond to the virus unless it gets that type 1 interferon tone and signal from the microbiome. So that's just so important to note, to keep in mind how critically important the microbiome is. It's not that the microbiome helps the immune system or increases the function of the immune system. The microbiome is part of the immune system, and it's intimately connected, where the immune system would not function if it wasn't for the microbiome. That was the first claim I made when I first started the thing, that I would want to be able to demonstrate to all of you that the immune system would cease to function without the microbiome, and this is just one reason why. And then on top of the commensals, being able to direct, activate, provide the threshold signals, recruit immune cells to sites of action, besides doing all of that as a facilitator of immune response, there are a number of commensal bacteria that have their own direct antiviral capabilities, right? So for example, my favorite genus and species, Bacillus subtilis as a commensal organism can produce a compound, a surfactant, that the study shows that it prevents the invasion from specific types of coronaviruses known as transmissible gastroenteritis virus. And this is, of course, not the SARS-CoV-2, so not to be misleading about that. But we know that commensal Bacillus surfactant can inhibit the transmission of this gastroenteritis virus from entering the intestinal epithelial cells. So that bacteria itself has a direct impact on the virus, not even through the immune system. We also know that there's anti-influenza activity of Bacillus subtilis based on some of the compounds it makes. In this case, it's a compound they call P18, which can completely neutralize influenza virus in vitro. Other studies have demonstrated that this strain that this strain has this functionality in vivo as well. So there's compounds, postbiotics, that are made by this type of commensals that can directly stop the virus. Here's another example. Bacillus subtilis produces an antimicrobial lipopeptide containing surfactant and fengicin, which is a strong antiviral compound. And this compound has been shown to effectively inactivate viruses such as pseudorabies virus, porcine parvovirus, Newcastle disease virus, IBDV. All of these different types of viruses can be neutralized by these single compounds being made by these commensal bacteria. And then, of course, there are antiviral leavens made from bacillus as well that are found in honey, in natural honey. And these leavens have been shown to be able to inhibit various forms of adenovirus, including respiratory RNA viruses like H5N1 and enteric adenovirus type 40. This in part gives honey some of its good immune properties because it contains these types of compounds. But the healthy commensal bacteria, the good, diverse, healthy commensal bacteria, are not only directing the immune system, controlling it, activating it, and so on, but they themselves are directly going after the pathogens with a number of chemistries that they can produce. And we also know that when the microbiome is compromised, the immune system doesn't function the way it should, right? We know this because there's lots of studies on antibiotics and the impact that it has on the immune system. For example, antibiotics found to weaken body's ability to fight off disease. Antibiotics weaken flu defenses in the lungs. This is a serious concern because I know here in the US, a significant number of antibiotic prescriptions are being written for things that the antibiotic won't help, right? And in some cases, it's really kind of a business pressure that the doctors feel. So we have a lot of immediate care centers here when people typically tend to try to visit immediate care centers when they're all of a sudden they wake up and they don't feel well, right? Because it could take 2-3 weeks to get in to see your actual doctor. So they go to these immediate care centers, which are of course staffed by emergency trained or triage trained type of doctors. And the doctors, even though they understand that the patient is likely suffering from a viral cold or flu, they are pressured into giving the patient an antibiotic or pill, which will make the patient feel like they're doing something to improve their health, right? Or doing something to improve the sickness so they can get back to work, not take so many days off, and so on. So the clinics that have a propensity to tell the patients when it's a viral infection that they don't need anything, they just need to go and rest and hydrate and take care of their body, those clinics don't get as much traffic as the clinics where they will write prescriptions for almost anything, right? So this becomes a big issue because if someone comes in and has the flu and is truly infected by the influenza virus and they get an antibiotic because the patient wants it to feel like they're doing something and the doctor is willing to give it, that antibiotic can actually reduce the patient's ability to deal with the flu and actually make things worse and maybe even drive up some secondary infections. And so we know that there's all kinds of damage to the function of the immune system if you take an antibiotic, or if your microbiome is damaged. The antibiotic is just a great way of illustrating microbiome damage and changes. Now, another area where this point has been extensively studied is in the area of cancer immunotherapy, right? So when we look at patients who undergo cancer immunotherapy, we know that the checkpoint therapy that they utilize really is trying to enhance your own T cells to go after the tumor. And if it works, the beauty of it is that there's really little to no harm, as I understand it. I'm, of course, not an oncologist and have not done this before, but in speaking to oncologists that we work with, from what I understand, the immunotherapy, if it works, the side effects are minimal. There's no damage to the host so much. And because it's your own T cells going after it, you may even develop some long-term immunity against that type of tumor, which means your immune cells have a better chance of stopping it. But what they found in a number of cancer immunotherapy studies is that if a patient is treated with antibiotics prior to starting the cancer immunotherapy, that it just doesn't work the same, right? So this key study here concluded that the gut microbiota plays a critical role in the anti-tumor immune response. This is there's increasing data showing that antibiotics change the composition of the gut microbiota and affect the efficacy of the immune checkpoint inhibitors. Therefore, the findings of our meta-analysis, and again, this is a meta-analysis paper, indicated that antibiotic use is negatively associated with outcome measures and with survival in cancer. And those are in patients treated with ICI immunotherapy. So really important way to illustrate how if you create dysbiosis in the gut, your immune system is simply not going to function the way it should. Now, what's really important to note is we've talked about how the immune system gets signals from the microbiome, either to recruit it to certain areas of action or to activate the microbiome, to provide that activating tone to the microbiome. And it's really important to note that all of those signals that the microbiome uses to communicate with the immune system are all inflammatory markers. Right, the microbiome will either directly release or cause the release of things like interleukin-6, interferon gamma, IL-12, IL-1 beta and alpha, interferon beta, TNF alpha. All of these are quintessential inflammatory markers in the body, and that's the communication that the microbes use to alert and track and recruit the immune system. So keep that in mind, because that becomes important when we start talking about many of your patients that may have chronic conditions. In the US, at least 50% of adults have one chronic illness, and at least 25% of adults have two or more chronic illnesses. And most of these chronic illnesses are lifestyle-related disorders, things like heart disease and Alzheimer's and Parkinson's, diabetes, obesity, and so on. And all of these conditions are indicated with chronic inflammation, right? Diabetes, cardiovascular disease, cancer, IBD, arthritis, autoimmune conditions. These are all driven by chronic inflammation, and they all have chronic inflammatory cytokines at the crux of the pathology of these diseases. And it's all the same type of cytokines and interleukins and all that. It's IL-12, it's IL-6, it's TNF alpha, it's interferon beta. All of these types of markers tend to be elevated in people with most of the chronic illnesses, right? So that's an important connection. So remember, the microbiome communicates with the immune system using inflammatory markers. People with pre-existing chronic illnesses tend to have elevated levels of these inflammatory markers. Right? So that becomes a really potent explanation for why it is that somebody with hypertension or somebody with diabetes or somebody with cardiovascular disease would have a 7 to 10-fold increased risk of dying from something like the coronavirus, the COVID infection, versus somebody that's age matched that doesn't have those comorbidities. Right? What is it about those chronic conditions, and what is it about those that predisposes people to having a much worse response against the virus? Is it the diabetes itself, or is it an underlying factor within that actually is part of being diabetic that compromises the immune system? So this area of lost signal can really help explain that. So think about the invading pathogen as a fire, as an actual flame of a fire. And then the smoke that the fire creates is the disruption in the ecosystem that is created by the presence of the pathogen. Now, the microbiome acts like a smoke signal or a smoke detector, and the microbiome is really keen on picking up the smoke from the fire to identify that there is a fire going on, right? And like any good smoke detector, once it notices smoke, it should sound an alarm. And in the case of the microbiome, the alarms are these inflammatory cytokines, IL-1, IL-6, and so on. And so like in this analogy, once the fire alarm is sounded, then the firefighters know where to go, make their way to the fire and help put it out, right? So this is how kind of the immune system with the microbiome functions. Remember, the invading pathogen is a fire, the disruption of the ecosystem is a smoke, the microbiome is a smoke detector, and then it causes the release of cytokines, inflammatory cytokines, like a good fire alarm, and then that recruits the immune system, which in this case are the firefighters. Now, let's say you've got chronic low-grade inflammation because of one or more chronic diseases, and that individual gets infected by a virus. Let's see what happens, because in this case, when you have chronic low-grade inflammation, you have those same signals that the smoke detector is trying to put out to recruit and attract the firefighters. Those same signals are going on everywhere in the body, right? It's chronic low-grade inflammation. It's everywhere in the body. IL-6 is elevated, IL-12 is elevated, IL-1 alpha and beta is elevated. Those are all markers that are elevated in circulation. Now, let's say the invading pathogen comes in, the smoke detector or the microbiome detects the smoke, sends out the signal to try to recruit the immune cells, and that signal gets lost because there are thousands of signals everywhere in the body. So the immune system cannot figure out where to go and what to actually go after, right? So then any invading pathogen gets the advantage of having more time to replicate and infect more cells before the immune system can finally find its way to that site of action. And again, it's because the immune system is completely distracted with all of these other alarms going on all over the place. So that issue of lost signal is a very significant issue. And that's one of the key explanations of how these lifestyle-based chronic disorders predisposes people to really poor immunological responses, especially when something like a new pandemic virus comes in, right? These individuals have compromised immune responses because their immune system cannot be appropriately alerted to the presence of a new infection. And so by the time the immune system actually notices the infection or gets the right signal to get there, it's been a longer period of time, which means the virus or the bacteria or whatever is infecting can replicate more. And now when the immune system finally shows up, the problem is a much bigger problem than it was to start, because the virus or the bacteria had more time to replicate and create more of a nuisance. Now the innate immune system has to come in and really create a robust response against it. And oftentimes that leads into things like the cytokine storm and damage from the immune response itself. Now there's a number of factors that can also compromise the immune system directly, things like Epstein-Barr virus infection or herpes simplex virus or cytomegalovirus. They infect T cells, they infect monocytes and macrophages, and Epstein-Barr virus infects B cells. And of course, chronic inflammation can drown out immune signals. And at the end of the day, the biggest driver of chronic inflammation that is creating the type of inflammatory response throughout the body, and that's driving this lost signal issue, is lipopolysaccharide. So when the gut lining is damaged and gram-negative bacteria, LPS, is allowed to translocate through and enter into circulation, and when you have elevated levels of LPS in circulation, that is one of the most potent drivers of chronic low-grade inflammation. And so the reason it can do that is because the way the body binds and deals with LPS is through a protein called LBP, which is LPS binding protein. It's produced by the immune system and it's circulating around the body all the time, specifically looking for LPS. Once it binds LPS, the combination fits onto a receptor called the CD14 toll-like receptor, TLR4 receptor on dendritic cells and macrophages. Once that complex binds that receptor, it causes the release of NF-kappa B, which then stimulates the release of a whole bunch of other inflammatory cytokines. So as you can see, if you have this pathology going on in your body all the time, you're constantly releasing NF-kappa B and interleukin-6, MCP1, interleukin-1 beta, and so on, right? Your own immune cells are constantly triggering inflammatory responses throughout the body. So you've got this chronic low-grade inflammation happening. Now, the clinical manifestations of this chronic low-grade inflammation are everything under metabolic syndrome. Everything is associated with LPS translocation and then the associated chronic low-grade inflammation, right? Every one of these conditions are. And in fact, studies in this case by American Diabetic Association have shown that when you look at the underlying pathology, the primary insult, as the authors call it, that start the process of the induction of diabetes, that comes from gut-derived endotoxins being present in circulation. And that activates an inflammatory state contributing to metabolic disease, right? So they've concluded that metabolic disease, like obesity and diabetes, is induced by chronic endotoxemia. Here's another important study in this topic, the CardioPrev study, which was 460 patients over 60 months. They followed these individuals who were pre-diabetic or at high risk for diabetes, and they followed a number of biomarkers to see which biomarker was the most predictive of the development of type 2 diabetes. And there was really only one, and that was LPS plasma levels as a biomarker to indicate the risk for type 2 diabetes, right? So LPS in circulation, the inflammation associated with it is driving things like metabolic syndrome, like diabetes, cardiovascular disease, polycystic ovarian, and everything else you saw. LPS can even create something called central insulin resistance by making its way up to the brain and causing inflammation in the hypothalamus, where now your brain can no longer read your blood sugar levels. And this is irrespective of body weight. And even if your pancreatic cells are working just fine, your brain cannot read and understand your blood sugar levels in order to cause the release of either insulin or glucagon and so on. We also know that the LPS in circulation is a key metabolic, the molecular link between cardiovascular disease and obesity, because these are the big questions, right? Why is it that somebody with high blood pressure, cardiovascular disease, why would they have 7 times the mortality rate on COVID versus somebody that's age matched that does not have those comorbidities? And so this kind of research paper provides a molecular link for that, showing that the key molecular link is the excessive amounts of LPS in circulation that upregulates inflammatory responses. And again, there's no surprise that people with high levels of circulating LPS, thereby having these chronic conditions like cardiovascular disease, diabetes, chronic respiratory disease, hypertension, all these individuals had 10, 7, 6, 5 times higher death rate than people with non-existing conditions, right? So if you've been asked and you've been thinking about how to explain it to your patients as to why someone with diabetes would have 7 times higher death rate with COVID than somebody who's the same age but does not have diabetes, it's because that individual with diabetes already has chronic low-grade inflammation. And that chronic low-grade inflammation is going to be the thing that creates a huge interference in the ability of the immune system to respond to the presence of a pathogen. Now, we saw this disruption within the microbiome as it directly relates to COVID risk. But here's something really interesting. SARS-CoV-2 spike protein can bind to E. coli LPS in human cells, right? So if you have elevated circulating LPS levels, it's plausible that that's another way the SARS-CoV-2 virus enters into human cells. It utilizes LPS as a shuttle to be able to do that. Now, this wouldn't be the first time that this has been shown with the virus. There's a number of viruses that have been shown to be able to utilize LPS as a shuttle to get into our cells and then cause infection. So this is a really important point to note, because not only is that LPS creating that low-grade inflammation, which creates loss signaling and a significant amount of time for the virus to replicate, it's also potentially driving the virus into the cells itself. And here's another paper that looked at metabolic endotoxemia, and maybe that is a big reason why obese individuals have a much worse time with COVID-19, because again, metabolic endotoxemia is a high level of LPS in circulation. Here's a study on obesity and COVID-19. This study is a systemic review of COVID-19 related to obesity. They looked at 75 studies. Results show that individuals with obesity had an increased risk for COVID-19. Obesity can, of course, cause insulin resistance, elevated serum glucose. You guys know all of that. What they stated in the article is that the pooled analysis shows that individuals with obesity were more at risk for COVID-19 positivity and then individuals that were age matched that were not obese, right? And again, what is the connection between obesity and compromised immune response, it's chronic low-grade inflammation driven by intestinal permeability and LPS endotoxemia. Here's another study on COVID-19 and dysbiosis. What they found is that the individuals that had the worst go and high levels of hospitalization, even mortality with COVID-19, tend to have a low level of Faecalibacter prosnitzi, which is a really critical keystone species. And they also tend to have low levels of bifidobacteria. And those decreased levels of bifidobacteria and Faecalibacteria itself can be found in individuals that became hospitalized at a much higher rate than individuals that were not. Right, so there is a correlation and a relationship between certain microbes in the gut and their presence and your risk of developing and being hospitalized with COVID-19. Keep in mind that Faecalibacter prosnitzi is one of those really important keystone species that produces a lot of short chain fatty acids, which are really key anti-inflammatory compounds. So this organism actually protects against inflammation in the mucosa, and it's been shown in numerous studies to be at low or no detectable levels in people with inflammatory bowel disease. So this is a strain that protects against inflammatory bowel disease. Same thing with bifidobacteria. They found a negative correlation with COVID-19. Bifidobacteria is, of course, found in the large bowel, and bifidobacteria modulates immune response quite a bit. Here's a second study. They were looking at a bigger cohort of individuals to investigate the gut microbiome changes of patients with SARS-CoV-2 infection during a hospitalization stay. And what they found that COVID-19 patients displayed an increase in opportunistic fungal pathogens like Candida albicans, Urus, and Aspergillus flavus compared to test controls. So test controls did not have the same level of increased opportunistic fungal pathogens. Aspergillus flavus is known to cause respiratory illness, and 53% of the COVID-19 infected subjects exhibited a continuous gut diversity change, meaning the microbiome diversity would change over time as the infection progressed, right? So we know that there's some sort of tie between the gut microbiome and your risk of a bad infection. And then here's another example of a study. The results showed that the gut microbiome composition was significantly altered in patients with COVID-19 compared with non-COVID-19 individuals, irrespective of whether patients had received medication, right? So just the presence of infection alone significantly altered the composition of the microbiome, or that patient could be infected because of a pre-existing compromised composition of the microbiome. So what they also showed is several gut commensal bacteria that are known to have immunomodulatory, in particular anti-inflammatory response, like Faecalibacter prosnitzi, Eubacterium rectalis, and bifidobacteria, were all underrepresented in patients and remained low in samples collected up to 30 days after disease resolution. So again, a second separate study confirming the same thing. Moreover, this perturbed composition exhibited stratification with disease severity and concordant with elevated concentration of inflammatory cytokines, right? So disease severity could also be predicted by similar measure of dysbiosis within the gut microbiome, and blood markers such as C-reactive protein, lactate dehydrogenase, aspartate, and so on were also elevated. Furthermore, they concluded that the gut microbiome dysbiosis after disease resolution could contribute to persistent symptoms, highlighting a need to understand how the gut microbiome microorganisms are involved in inflammation and COVID-19. This is referring to the long hauler syndrome, and there's some other studies that have come out since that show that long haulers tend to have immunological profiles of people with severe chronic illness with chronic low-grade inflammation, right? So, and people with autoimmune conditions, for example. So, having a healthy microbiome becomes absolutely critical for the function of your immune system. And of course, one of the most important ways and easiest ways of reducing LPS, endotoxemia, and the related inflammatory markers is using the spore-based probiotics. We've published on that back in 2017. We're actually doing a study that's 4 times the size right now and 3 times longer. So we're quite excited to be able to share that, so we can share some additional information. But looking at the initial impact that the probiotic had on LPS levels postprandially, this is what a standard curve looks like. At time 0, people come in fasted. You measure blood LPS levels, and then when you feed them a meal here at time 0, you can see if they have severe endotoxemia, how rapidly within 5 hours the level of LPS in serum dramatically increases. 5-6-fold increase is what we're looking for. So we recruited a number of patients that had this severe endotoxic response, and then we gave them the probiotic for 30 days, and this is what their response in 30 days looked like. Basically non-detectable or very, very low levels of LPS found in circulation, even with individuals that had this massive, robust LPS response just 30 days prior. So just putting them on the probiotic had that huge impact. We saw it in a larger cohort as well, a 60% reduction in the amount of LPS that's in circulation after 30 days of taking the probiotic. And the scary thing is the placebo group not only didn't go down, but they increased by 32%. And so I think I mentioned the heat map earlier. So this is a great way to kind of visualize the overall inflammatory status of the spore-based individuals versus placebo individuals. Of course, using a heat map, the more red it is, the more intense the number is, a bigger increase from baseline. So as you can see, the spore treatment individuals were having overall reduction in inflammation, whereas the placebo people actually increased their inflammation on a regular basis in that 30-day period. And these are all the levels of those key cytokines we talked about: interleukin-6, interleukin-8, interleukin-1 beta, and so on. And so understanding that stopping LPS from translocating from the lumen of the gut into circulation becomes a really important part of stopping that chronic low-grade inflammation, right? Or at least reducing that chronic low-grade inflammation. So the spore-based bacteria, spore-based probiotics can take care of that part, right? But then there's also these characteristics of a healthy microbiome as it relates to immune response, things like improved Faecalibacter prausnitzii, increased levels of bifidobacteria, increased production of short-chain fatty acids. The big question is, how do we achieve some of those other things? So we've been doing studies on synbiotics, which is taking the spore-based probiotic and mixing it with an oligosaccharide prebiotic. So we use fructooligosaccharide, galactooligosaccharide, lactooligosaccharide, and we look at the synergy between the oligosaccharides and the probiotic bacteria. And what we found in some of our studies is that the ability of the symbiotic to increase the concentration of keystone species like Faecalibacteria is really quite mind-boggling. This, as you can see, is a 2-log increase from baseline levels of Faecalibacteria in these microbiome samples. And so what we see here is a 100-fold increase in the protective Faecalibacter prausnitzii. Same thing in bifidobacteria as well, very significant increase. Lactobacilli, Akkermansia. So across the board, we see this significant increase, which is really important because, as we know, having high levels or healthy levels of Faecalibacteria, Bifidobacterium longum especially, Akkermansia, Lactobacilli are all highly protective and associated with really good, robust immune response. So in conclusion, a healthy, diverse microbiome provides critical signaling and energetics to the immune system to elicit proper immune function. Higher pathogen loads disrupts immune response because when you have opportunistic organisms in your commensal flora at high levels, what you tend to see is the opportunistic organisms do not signal the immune system in the same way that the good commensal bacteria do when they notice an infection, right? Because the opportunistic are actually looking for the opportunity to raise their virulence factors. And so having a microbiome with low levels of opportunistic organisms, high diversity, high levels of protective organisms like Faecalibacteria, Akkermansia, and so on, though that becomes the biggest critical factor in how healthy your immune system is. We know that a disrupted microbiome is also the most prevalent source of chronic low-grade inflammation through endotoxemia. We also know that immune support ingredients, things like vitamin C, vitamin D, zinc, as important as they can be, they cannot overcome the dysfunction of the immune system as it relates to the dysfunction of the microbiome. You simply cannot take enough vitamin C to overcome how a dysbiotic microbiome will compromise your immune system, right? So you need to address the microbiome while you're taking the micronutrients so that the micronutrients can be utilized properly. The success of preventative measures like vaccines, for example, to elicit an immune response will depend on one's immune capabilities and therefore one's microbiome. So for the vaccine to be more effective, we need to ensure people's guts are healthier so that their immune system can mount a very appropriate response. And simple measures can make a big difference in improving your gut microbiome. First is diversifying the diet, right? More different types of food should be consumed on a daily basis, and new types of food should be added on some sort of consistent level. Lower stress. Stress is the biggest driver of epithelial, of intestinal permeability, and it's the biggest driver of opportunistic organisms increasing their virulence and their prevalence within the microbiome. So stress is a huge driver of chronic low-grade inflammation. Getting outdoors makes such a big difference. There are numerous studies that show that when you engage in the outside environment, especially in natural environments, not so much walking up and down the streets of the city, but if you can go on a hike to a park or the woods or somewhere at the beach, somewhere like that, then that interaction with nature has a huge impact. Using a spore-based probiotic really can make a big impact, and that's a simple impact. Focusing on leaky gut solutions, because we know leaky gut is driving the vast majority of chronic low-grade inflammation, bringing down inflammation through the production of things like short-chain fatty acids and so on. Prebiotics like oligosaccharides can be quite useful. We've shown how compatible they are to the spore-based probiotics. And then some of my favorite things to take in, polyphenols and omega fatty acids can be really powerful support tools, because both of those get metabolized by the microbiome into anti-inflammatory compounds. So they really help with bringing down inflammatory damage. So that's the end of my talk. Thank you so much for being with us today. And I'll leave you at this slide. I don't know if we have time for questions. I know I went over a few minutes. I'm sure people have things to get to, but I'm happy to take some questions if there are any.
Thank you, Kiran. We're opening the floor for any questions. You may unmute yourselves, or you may also show your face if you like.